KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
Neet, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Hi students, good morning to all. So in this video we are discussing allelic interaction or intragenic interaction. This particular allelic interaction or intragenic interaction, so we have completed one example in previous video. So in one previous video, what is information we have seen in allelic interaction? So I have given information regarding incomplete dominance. So in incomplete dominance, flower color of Mirabilis jalapa and Antirrhinum and Andalusian falls. So likewise, a few examples I have given. So in this video, so I will give information regarding dominance. So that dominance also not completely co-dominance. So now in this video, definitely I will give information regarding co-dominance. In this phenomenon, one important point, what is that? No, codominance. Codominance also comes under allelic interaction or intragenic interaction. Okay, so now let us see in this kind of a phenomenon. So both the genes are express a particular character in F1 hybrid progeny. On one line, by reading of first line only, so entire concept we understood. What is the particular point in this phenomenon? Both the genes express for a particular character. So in this particular case, no, definitely we are understanding one point. Both the gene, so okay, both the gene expresses. Here try to avoid the S. So both the gene express a particular character in F1 hybrid progeny. So means definitely what is the thing? What is the gene consist of both the different types of alleles? Both will be expressed. Okay, so there is no blending of characters. Means here what are the characters of alleles are there now? So both characters without any blending, without suppressing any other, no. So both will be expressed. There is no blending of characters where they both characters expressed equally. So here there is no domination. So that's why there is no blending of characters. So why? Because whatever the alleles are there in the gene, no, so all those alleles are mutual interaction. You don't suppress me, I won't suppress you, I don't suppress you. So likewise, so like a friend's madri, so both will express at a time. The friendship madri. So what are the alleles are there? Dominant allele, recessive allele, and the madri rendume both are not dominant and both are not recessive. So both are friends madri. Okay, so likewise here what is the point? In codominance, in this kind of phenomenon, both the genes express a particular character. Both the gene express a particular character in F1 hybrid progeny. There is no blending. There is no blending of characters where they whereas both characters expressed equally. For examples, so definitely where we can see codominance is seen in cattle for coat color. Okay, so in cattle, no, coat color. So coat color we can observe. So this kind of codominance best example. But here somewhat examples we have seen. So that are examples also we will discuss, no worry. But in Neet examination point of view we are discussing, no, that's why along with our textbook A example, some other examples also I brought for you. Next one. Now when a black parent, now let us take the example, when a black parent is crossed with white parent, where in cattle means in a simply we can take this particular explanation in animals. So simple point of view we can take in animals, no, black parent, one black parent is there, that particular one when we are crossing with white parent, so a roan color F1 progeny is produced. The combination between or the mating between black and white if you do means, so roan color means the mixture of or which one, black and white color. So that one only we can be taken as roan color. So is produced in F1 progeny. Or roan color offspring we are observing in F1 progeny. So when we obtain F2 generation, so let us move forward. So when we obtain F2 generation, what we are doing here from the F1 generation, so the ratio of black and black white, once you see that black and white roan in brackets for easy to understand, I have written like this, black and white combinedly we will be calling as roan color. Okay, now if black one is there and black and white combination roan is there and white color cattle also obtained in 1:2:1 ratio. My dear students, in this video, so in this particular concept, what is the point we are understanding? No, so even in codominance also, even in codominance also we are obtaining the, so here 1:2:1 ratio as like in complete dominance as in monohybrids Mendel's or as in Mendel's monohybrid cross genotypic ratio. So thereby here 1:2:1. So in how many cases we are observing means, so we have seen a genotypic ratio in a monohybrid cross we seen, and incomplete dominance means there both genotypic characters as well as both phenotypic characters. So definitely we have seen the presence of we obtained 1:2:1 ratio. Now even in codominance, even in codominance also we are getting. So even in codominance also we are obtaining, what we are obtaining, definitely we are obtaining the 1:2:1 ratio. In 1:2:1 ratio so black one and roan color means black and white mixture rendu and white color one. So that's why here 1:2:1 ratio. One important bit is there, sorry, note we have written one point, listen there. So in F2 generation, the combination of F1 and F1 self crossing one, so definitely we will obtain F2, no. So in F2 generation is obtained in animals by sib mating cross, means the crossing between siblings. Means what is the information now? So when these offsprings, offsprings of one parent is there, no. So here what are the offsprings here we have been taken, no. Those offsprings must have single parent. From one parent, suppose over 4 offsprings has come means, so if mating between these 4 offsprings is. Okay, point you understood. So thereby actually brother sister of sister madri. If brothers or sisters are there, no, between them. Likewise animals, in animals, so suppose if you do crossing, so such kind of crossing we will be taking as a sib mating cross. Okay, so when we will be doing like that, what is happening in F1 generation? Once let us see this one. First of all, we have taken black parent and white parent. So black parent let us take as R1 and R1, and white parent let us take it as R2 and R2. Now here in a gametes point of view, here R1 one gamete and here R2 as another gamete. The combination of these two, R1 and R2 as F1 generation. So we know very well, we have seen so many times. Now for obtaining of F2 generation, what we are doing in animals, sib mating cross we are taking. Sib mating cross is nothing but, once you see Sibmating cross is nothing but the mating between related offsprings, means half sister, half mother, half brother, likewise. Means whatever the offsprings are there, no, all these offsprings must come from the same parent. So now in this particular situation we are crossing between these offsprings. Means definitely blood relation is there, no, so between them only we are crossing. So thereby F1 into F1 generation, when we are going to do sibmating cross, no, F1 into F1 means R1 R2 into R1 R2, isn't it? So thereby here one R1 R2 offsprings in the side and another ones in the side. So one we will be considering as male and another one definitely we must take it as a female one. R1 with R1, so R1 R1, R2 with R1, R1 R2 and R1 with R2, R1 R2, next one R2 with R2, R2 R2. R1 R1, so black, R1 R2 roan color. So here offspring is there, no, so this particular R1 R2, so the mixture of both parents, so thereby roan color, black and white color. Next one here R2 R2 is there, no, that one is white color as we have seen. So means this particular information, if you see there, no, this particular information, so we can remember monohybrid cross. So monohybrid cross we can remember why, because there R1 black parent, like R2 white parent, but here in F1 generation point of view incomplete dominance we can remember. Why? Because here why we can be taken incomplete dominance here both parental characters are not there, but here another one new color came, no, so black and white mixture came. So later again as we have seen in incomplete dominance, no, here also we obtained 1:2:1 ratio. 1:2:1 ratio even in codominance. So this is some more information related to the like cattle color or cattle coat color information. So by relation of the codominance. Next one special note is that. So what is a special note or what is another extra information to be discussed means in incomplete dominance, idi 4 lines are there, 4 lines nalla understand pannikonga. Now in incomplete dominance characters are blended phenotypically. Incomplete dominance nalli, so definitely here the characters blended means here one allele is there, no, that one is like suppressing the another one, so thereby fighting is there between the each and every one. So but here so where in codominance, no, we have seen like that, sorry, in incomplete dominance we have seen blending of characters phenotypically we see, but in codominance both genes of a pair, one more time you see there, both genes of a pair exhibit both characters side by side. Means in this particular case, no, what are the main important points are there in codominance, no, even though all these are all present together, so here in a way while in a codominance, no, both the genes of a pair exhibit both characters side by side, side by side, and effect of both characters is independent from each other. So means here both characters or the both like what are the pairs are there, no, in one particular gene, no, all those are all being independently and showing their own characters. So thereby here what is the best point here we can take, no, so in our examinations point of view textbook information we have learned, no, so there when IA with IB is there, no, so there what is a blood group formation we can see AB blood group, means here even though A and B are present together, so here any one character is not expressed here, both characters are expressed there. Point you understood. Thereby here what is the blood group of particular thing we are observing is AB blood group we are observing. So thereby my dear students, so one important point mattum nalla understand panikonga, what is that, no, in incomplete dominance all characters are blended phenotypically, so blended phenotypically, but whereas in a codominance, no, so without any blending, so what are the characters even though present together side by side, no, without any suppressing nature, both characters are expressed out in independent each other. Point you understood, so that one, so that's why while explaining codominance, ABO blood groups we will be taking. So in ABO blood groups, AB blood groups are definitely showing off codominance nature. AB blood group inheritance, so there IA with IB definitely showing off AB blood group. So A and B are present together. Even though A and B present together, both characters are expressed. And one more example, carrier of sickle cell anemia. So there also HbA and HbS. So this particular example also we can be taken under codominance. So we can be taken under codominance. Next small concept, another concept, that is the third one under allelic interaction. So that is allelic interaction. So there multiple alleles. Okay, multiple alleles point of view, this particular concept also is important. So more than 2 alternative forms of a gene are called as multiple alleles. One more time you try to see. So what is the point here we are discussing? More than 2 alternative forms of a gene. One more time. This by heart pannikonga. Sometimes even in examinations, so this one they are asking as one definition. Okay, more than 2 alternative forms. Actually, suppose one gene is there, means there 2 alleles only must be there. But here how many are there? More than 2 are there. Point understood. Suppose one gene if you take there, there 2 contrasting alleles till now we know. But if one gene nalli how many number of contrasting alleles we are going to study? More than 2. Renduku mele da iruk. Okay, so that kind of renduku mele alleles irukana means more than 2 are there, means definitely we can be taken as a multiple. Many are there means we will use a multiple word now. So that is the information. More than 2 alleles of forms A or forms of same gene are called multiple alleles. I hope you understood this one. Now these multiple alleles are formed due to the mutation, or due to the process of mutations. So or due to the mutations means sudden changes which occurs in chromosomal sequence. So these multiple alleles are definitely are formed because of the mutational processes. So next one, multiple alleles are located on the same locus of homologous chromosomes. So suppose if you take there where the particular gene on the chromosome, that particular location only we are calling as locus. Locus means purinjita. One more time you see there, locus is nothing but gene location. Gene location where the gene is present on the chromosome, that particular place will be there. That particular place on the chromosome, for our convenience to study, we have given one name as locus. So definitely multiple alleles are located on the same locus of homologous chromosomes. So homologous chromosomes means suppose if the chromosomes are morphologically, structurally like seems to be same, means so such kind of chromosomes we will be taking as homologous chromosomes. Next one, if one line like mathematically use once you try to take this one. So if n is the number of allele, one more time, if the n is the number of allele of a gene, then the number of different possible genotypes. So to find out the number of possible genotypes, so what is a formula here we can use means, so definitely we can use one formula here to find out the possible number of genotypes. Don't forget to find out the possible number of genotypes. Now one formula we are going to learn, that for particular formula is n into n plus 1 by 2. One more time by heart pannikonga, n into n plus 1 by 2. This particular formula we can use for the finding of total possible number of phenotypes. Okay, so next one, example of multiple allele. Example of multiple alleles there. First one, ABO blood group. First one there, ABO blood group. If this particular ABO blood group we can use for the codominance, but there completely ABO we are not using, just we are taken what we have taken, we have studied the example AB under codominance. Even though we are taking ABO blood groups, but there where we will study the presence of codominance only in between the A and B only, no. So that's why here in a previous example I have taken only AB blood group instead of ABO blood group. I hope you understood. One more time you listen. Why in codominance, why I didn't take ABO blood group and why I have taken AB blood group means codominance exclusively we can see in between of A and B only. That's why, so even the possibility is there to take ABO, but why Raju sir took only A and B means codominance we are observing between the A and B only, not between the or not in between the ABO blood group. Point you understood? That's why AB blood group of inheritance I have taken example for codominance, but here in multiple alleles now definitely I need the help of ABO blood group for their explanation. Point you understand பண்ணிக்கோங்க. ஸோ கொஞ்சம் டீப் இன்ஃபர்மேஷனுக்கு போகும்போது ஒரே டைம் எக்ஸ்பிளைன் பண்ணிக்கு முடியும், பட் அண்டர்ஸ்டாண்ட் பண்ணிக்க கொஞ்சம் ப்ராப்ளம் ஆகும். ஸோ அது கொஞ்சம் நல்லா பார்த்துக்கோ. நான் multiple alleles examples point of view, ABO blood group. So in ABO blood group example if you take there, no, ABO blood groups are determined by 3 number of alleles. Easy, the once you see that their total number of alleles, how many are there in blood groups only determined by 3 number of alleles. What are those? Very easily we can find out. What are the 3 alleles we can discuss? A, B and I. Simple this I da, not I mele 0 illa, I da normal da. IA, IB and I. இப்போ இது ரெண்டுமே dominant தான். And normal I, இப்போ small capital I, small I is there, no, small I மாதிரி இது write பண்ணணும். சரியா? இப்போ 3 number of alleles are there. அங்கே IA is dominant, next one IB is dominant, and I small I small I is recessive one. இப்போ one important point I will give you, so நல்லா சேவ் பண்ணிக்கோங்க. Suppose if you are crossing this IA with I character only, A blood group only will come out. Next one, if you are crossing with IB with I, no, B character only means B blood group only will varum, express out. இப்போ the combination of IA with IB yaar varum theriyuma, both varum. Why? Because that example only, no, pakatali example we have discussed under codominance. Understood, no? So that's why suppose if you are crossing with IA with IB, here the both are dominant, no, that's why both characters comes. If both character comes means, so that particular example we can be taken under codominance. Point you understood here. So now here possible genotypes. One more time, once you see there, possible genotypes. So what are the possible genotypes over there? Once you see there, A, B, AB, and one more is O. One more time, once you see there, possible possible genotypes. Sorry, possible phenotypes we have to write. Okay, man. Now once you see there, I changed this one. Now possible phenotypes. So now here the number of possible phenotypes. Phenotypes means what are the characters will be expressed out. So possible phenotypes point of view, how many are there? A, B, AB, O. So once you see there A, B, AB, O. Now 4 number of possibilities are there, no? Where under phenotypic 4 we are observing. So there possible genotypes number. இப்போ possible genotypes number point of view, இப்போ formula is for finding of a possible number of or possible genotypes number point of view, n into n plus 1 by 2 one formula we discussed, no, அது formula இங்கே apply பண்ணிக்கோங்க. இப்போ பாருங்க number of alleles how many are there? 3 alleles are there, no. இப்போ number மட்டும் போதும். Okay, இப்போ IA இருக்கு, IB இருக்கு, I இருக்கு. So இப்போ 3 alleles are there. இப்போ instead of n place or instead of n, so let us put 3. Why? Because 3 number of alleles only, no. So thereby 3 into 3 plus 1 by 2. இப்போ 4 into 3 12 by 2 is equal to 6 genotypes. Okay, so thereby 6 number of genotypes definitely we can find out in ABO blood groups. So thereby my dear students, one point only save பண்ணிக்கோங்க. அது என்னன்னா in ABO blood group in ABO blood group total number of alleles 3 are there, and total number of phenotypes of 4 are there, and total number of possible genotypic number is 6. Simple amma, total number of alleles 3 3, and possible number of phenotypes 4, and possible number of genotypes 6 podum enough. Okay, so that is the information regarding ABO blood groups. But ABO blood group, don't forget my dear students, if more than 2 alleles are there, no, more than 2 means 3, 4, 5, 6, more than 2 LMA we can be considered under multiple alleles. Okay, if more than 2 alleles we are observing at one particular gene location, no, definitely that particular phenomenon definitely we can be taken under multiple allele. The word side heading itself giving information. Paringa once multiple alleles means presence of more number of alleles or more than 2 number of alleles of one gene. So I hope my dear students you understood this. Next one, second example also let us take. There coat color in rabbits, there 4 alleles are there. Coat color amma, so coat color ana mele skin color mele konjam color will be there, no, definitely buffaloes means black color and cows means white color. Likewise amma coat color in rabbits, no, there 4 number of alleles. This example mattum podum. Inga 4 number of alleles where we are taking means coat color of rabbits. Okay, or coat color in rabbits, no, 4 number of alleles are there. 4 number of alleles are there means how many number of genotypes are there, you can guess. So thereby 4 into 4 plus 1 by 2 if you apply, no, so definitely, so total number of genotypes we can obtain in coat color of rabbits. This is easy da, so just alleles number mattum save பண்ணிக்கோங்க. Next one, இப்போ பாருங்க codominance we have seen in allelic interaction, no, codominance we seen in the side board part nalli we seen, no, ade. Next இப்போ we have seen multiple allele information. இப்போ new concept amma lethal gene. This is new concept, lethal gene. So what is the information? So what is the importance or what are the points to be discussed in lethal gene? Now let us have a look on this. Now gene which causes death of individual, amma paringara. So lethal gene na gene which causes death of individual when it comes in homozygous condition called lethal gene. So very important. So what is the point here we can understand means, so here lethal means lethal means kill or toxic or poison. Okay, so lethal means definitely, so because of the presence of this one definitely causes death. So means here what is the point? Gene which causes the death of individual when it comes in a homozygous condition. My dear students, when particular gene will be there in a homozygous condition, once I will show you that particular diagram, whether it is appearing or not, for me doubt is there. Once I will go there. Once here once you see small table we have drawn. First let me explain. Exact light spotlight now you see exact light spotlight ange yaar irukana capital Y irukama inge. Ipo now only what is the point we learn, what is the point here we learn. Now once you see there, go to the information. So here gene which causes death, suppose one gene definitely is responsible for death. Suppose in one individual when the gene comes with homozygous condition, capital Y comes means, so that particular thing, no, we can be taken. So that particular homozygous state of a gene, definitely causes death. That's why here with another ink I have written, once you see there, capital Y is responsible for death. Why we have written like that, theriyuma? Capital Y homozygous state, no, if homozygous state is there means according to the statement information definitely that particular organism must go death. definitely meet death. So thereby here that particular gene means what is the gene which is responsible for death of an organism? No, that particular gene we will be taking as lethal gene. Ipo that particular gene also when it causes a death only in case of homozygous state. Ange rendume means what are the both alleles are there? Now both alleles must be in same da. Appadena capital Y must be there. So that is important. Save pannikonga. Ipo lethal gene was discovered by so this particular kind of information who is a scientist discovered this one means discovered by Q0 L Q0 scientist name adu maadhirde irukku but save pannikonga. So Q0, so the lethal gene was discovered by CUENOT. Okay, Q0 in coat color of mice. In coat color of mice, only Q0 scientists discovered the lethal gene. So now information regarding lethal gene point of view. Yellow body color. Yellow body color in a brackets only capital Y. I think so in a video it is not appearing clearly. That's why through my voice you can save. Sariya? So right. Now yellow body color now we have taken as a capital Y was dominant over the normal brown color small y. Now what are the characters we have taken? Yellow body color capital Y and normal brown color Y we took. Now the gene of yellow body color is lethal. இப்போ பாருங்க the gene of yellow body color is lethal. So homozygous yellow mice are never obtained in the population. One more time you see that. So homozygous yellow mice are never obtained in a population. Ipo homozygous means capital Y dana. So this particular capital Y yellow mice definitely will not be obtained or will not be present in population. Why? Because our knowledge point of view now only we will learn. Ipo homozygous yellow character comes means capital Y is there means definitely that particular gene responsible for death. That's why homozygous yellow mice characters yellow mice will not be there in the population in a group of mice. Why? Because it dies in embryonic stage only. புரியுதா இப்போ so why sir இப்போ capital Y இருக்கனா sir why sir that particular mice yellow mice character we are not observing in a population of mice sir means because of the presence of homozygous capital Y color no. So that particular gene is acting as lethal gene so our knowledge point of view definitely that kills the death of a particular individual. Correct dana. So that's why here when yellow mice were crossed among themselves segregation of yellow and brown body color was obtained in 2:1 ratio. So now here the ratio of organisms last line ratio of organisms we have written that one as 2:1. Sorry to say that one is not appearing clearly in video but through my voice you can save pannikonga. Idi 2:1 sir ipo why sir we have taken 2:1 ratio means once you see that particular table here table is there no ange table nale what is the table we have taken information offsprings no capital Y small y we took. And in the side also offsprings gametes capital Y small y here one gamete and here also capital Y small y here we took. Ipo now total number of offsprings how many are there? 4 offsprings are there but first one is capital Y means definitely that particular offspring will die in only in embryonic stages itself. Embryonic stages only comes means it will not be delivered. So thereby when this particular capital Y character when it is got death in embryonic stages now progeny how many number of formation taking place how many numbers will be delivered out just 3 da ange. So ratio we can be taken like this 2:1 ratio. Why? Because ange here small y definitely will be brown bodied one and heterozygous yellow state yellow one. So but homozygous yellow color mice will not be born. Why? Because when homozygous capital Y are present together at the time that particular gene responsible for death of an organism. My dear students, you understood this one. Suppose it is a new concept now. Suppose if you are unable to understand no worry amma. So school will be opened in next month, I think so, next month or in the month of October, definitely at the time. So definitely one more time I'll explain. So what are the concepts you are unable to understand, so through these videos, no, so no worry. So such kind of or what are the concepts are difficult to understand in this video, so definitely I'll explain in presence of you, no worry. So of my subject, so definitely I'll give you entire information which is unknown to you. Okay ma, thank you and be safe at home.